Continuing to talk about thin layer chromatography, there are a few points that need to be clarified. That silica gel is the main coating material which is used, and it is the reaction of the dye molecules with the silica, whether it is adhering to the silica or not so adhering, will decide the polarity of the dye, and accordingly the polarity of the solvent will then start be chosen, and once the desired chosen solvent system is taken, the dye starts moving at a different RF. RF means the retardation factor, and that retardation factor for dye 1 or dye 2 will always be different in different polar solvents. So, therefore, the solvent polarity is of prime importance, and secondly the RF of the dye is of importance. It can be actually, because these are all colored molecules, they can be seen by the naked eye by the use of visible light, or sometimes these TLC plates are then developed in iodine chambers. So, they become brown spots, and one can actually see the dye molecule on the TLC. If it is a mixture of two dyes, it will show that there is an example that I have taken, that there was a mixture of red dye, and on one side I have spotted, and which is showing only one spot, whereas on the right hand side the spot shows two red colors, which means that the right hand side dye mixture, which was also a red dye, actually contains two components, and the left hand side dye is just a single component dye. Now, when I was telling you that the dye can either be seen visually, that is because you can see the red color. Otherwise, it is put in the iodine chamber and all the dyes appear as brown dyes or brown spots. So, that is what I meant. Now, going back to column chromatography are like tubular structures which have a stopcock at the end, and the column is filled up with silica gel. Of course, the silica gel that is used in this case is different from the silica gel that is used for TLC. So, because on the TLC a very even smearing of the silica gel coating needs to be done, therefore, it is finer in particle, but this silica gel which is used in column is slightly coarse, and once the silica gel is filled with the help of by making it into a slurry, then mixtures of dyes can be separated on them. So, that is the purpose of, but the basic fundamental of separation or identification is the same as what it is for thin layer chromatography. So, if I have to explain a little more in detail, in case of column chromatography, mixtures of components enters a column chromatographic process, and the different components are flushed through the system at different rates in different polarity of solvents. These differential rates of migration as the mixture moves over adsorptive materials provide separation. Repeated sorption desorption acts that take place during the movement of the sample over the stationary bed determines the rate. The smaller the affinity a molecule has for the stationary phase, the shorter time spent in the column. So, it is something which is valid for any and every organic compound. So, it is how these organic compounds are adsorbing and dissolving and adsorbing and dissolving on the column that the rate of elution will be decided. And because there is a long traversing area, the rate of traversing becomes larger and larger. So, they get separated, although it all starts with a mixture, but then gradually, because each dye has a different rate of traversing on the column. So, therefore, it moves separately, and when it moves separately, it would be dependent on the fact, the movement will be dependent on the fact that how nicely or how not so nicely it is having an affinity for the silica gel. And if it is having a good affinity for silica gel, it would be adhering to the silica gel, and therefore, it will not elute very fast, but other components will elute. So, therefore, one can come to a conclusion that it is all a process of sorption and desorption that decide the rate of its flow onto the column and finally, out of the column. In the analysis of natural products, column chromatography is specially advantageous as to purify individual chemical compounds from mixture of compounds. Two methods are generally used to prepare a column, the dry method and the wet method. In most applications, the stationary phase, either silica gel or alumina, which is mixed with the solvent, being used as a mobile phase to yield thick white slurry. The mobile phase is a liquid that is chosen to maximize the separation of the sample. This can be water or any other organic solvent. So, it is important that what kind of a solvent we should use, the same solvent in which the silica gel slurry has been made. And if the polarity of the slurry and the polarity of the solvent that will be subsequently used are very different, it will cause some kind of erratic movement on the column. Therefore, one should remember that one should not abruptly change the polarity. Gradually, the polarity should be changed, so that the rate of traversing does not alter. Different other chromatographic techniques were also used in colorant identification and isolation from plant species and will be described. So, this is one of the basic methods of separation if it is a mixture of dyes. Ultraviolet visible spectrometric method. This we will do in a little bit more detail, because this is one of the quickest and most easily doable kind of technique to identify the dye molecule. A dye is dissolved in suitable solvent system and scanned through UV visible spectrophotometer. Identification of dye by this method involves an empirical comparison of the details of the spectrum, that is, the maxima and the minima point of unknown with those of the pure compounds. A close match is considered to be good evidence of the chemical identity, particularly if the spectrum contains a number of short and well defined peaks. So, this is one of the best method as what I mentioned. Why? Because the lambda max is the main criteria, because every colorant molecule or every chemical molecule may have a visible or a UV region lambda max, and that would depend on its chromophoric group, and so it is a significant of its structure as well. Principles of UV. When a material is illuminated by light, specific wavelengths are absorbed depending on the molecular structure present. Electrons in the ground state molecule absorbing light energy and moving to an excited state cause this. The absorption intensity depends on the wavelength, and the absorption spectrum, that is the curve measuring absorption intensity, changes accompanying wavelength changes for monochromatic light illuminating a material. And it is a characteristic of a specific material. Analysis of material based on this principle is called absorptiometry. This analysis can be used for various purposes, that is identification as well as quantitative analysis. Right now, let us only concentrate on the identification, because we are trying to learn various techniques which can be used for the identification of dye stuff, be it natural dye or synthetic dye. So, the best way of doing that is that light or monochromatic light is passed through the dye or the dye stuff, and the lambda max is observed, which is a very characteristic of a dye. For example, I will tell you that AR18 and DR80 showed a lambda max of 509 and 543 nanometers respectively. By whichever UV spectrophotometer you take this or you evaluate the UV spectrum of these two dyes, the lambda max will always show this, which goes to prove that their structural details are such that the chromophore groups are in such a manner that it would absorb light related to these two wavelengths only when these dyes are present. So, that is how one tends to identify dyes, and this is valid for both synthetic dyes as well as natural dyes. The electronic state analysis. In addition, molecules absorbing light and entering the excited state later lose energy through thermal dissipation, collisions and other molecules or other processes and return to the ground state. These processes are called radiation, less transitions, and may include the emission of absorbed light energy in the form of light. Such re-emission processes include fluorescence and phosphorescence. Analysis using these phenomena is called fluorescence photometry. So, this is another kind of study where the glowing of the light is observed, and that is because it is not usually transferring to the ground state in a very normal manner. There are some other things that are happening along the process, and what is exactly happening, that there is some amount of radiation while this ground, when the electrons are coming back to the ground state, some radiations are given out, which causes fluorescence. Now, this is how the UV spectrophotometer looks like, and I just took a picture for you because you should be able to at least identify that this is a model of UV spectrophotometer. Now, when a spectrum is evaluated, this is a typical UV spectrum of synthetic indigo and natural indigo. Now, you will see that if our layman looks at these two peaks, these two lines, one indicating the synthetic indigo and the other indicating the natural indigo, you will say that more or less they look similar, but they are not really similar. What makes them dissimilar is the fact that the peaks at 285 as shown by natural indigo is slightly different in the case of synthetic indigo, and it shows at 288 nanometer, which is in the UV region, and in the visible region if you see, the natural indigo shows a peak at 603 and the synthetic indigo shows a peak at 607 nanometer. So, there are very small variations, but nevertheless with the help of UV itself, I told you with the help of TLC we could identify that whether there is a natural indigo or synthetic indigo. Similarly, with the help of UV machine, one can identify looking carefully at the lambda maxes. So, the lambda max at 288 and 285 are definitely different. Similarly, the lambda max in the visible region, that is 603 and 607 are different. So, they make these two molecules quite different from each other in terms of UV spectrum. Similarly, the next machine that we come to know or look at it, which is used for the identification of dyes, is Fourier transform infrared spectrometry. And the studies show that functional groups were identified by FTIR of purified dye extract. Major peaks were identified for different types of CC, CH, CO stretching and bending vibrations. Absorption in the infrared region is due to molecular vibration of one kind or another. The spectrum is generally very complicated and contains many absorption peaks. Nevertheless, there is a region which is for the interest of the analyst, and there is one region which is very specific, which need not be understood fully, but the other half of the spectrum gives sufficient information so that one can at least derive at some kind of conclusion. IR absorption spectrometry or spectroscopy. The IR absorption spectroscopy is based on the absorption of IR radiation by molecules. I am all along mentioning molecules because dyes are also molecules, organic molecules, and is most widely used for the identification of organic compounds. The atoms in molecules vibrate constantly in a variety of stretching and bending motions. The different types of motions called vibrational modes. Atoms that are connected by covalent bonds can stretch or bend at natural resonance frequencies, which depend on the strength or stiffness of the bonds. So, a single bond C will show a vibration mode at 1200 centimeter inverse. Carbon-carbon double bond will show at 1650 centimeter inverse, and carbon-carbon triple bond will show at 21 or 2200 centimeter inverse. Now, you see that if we only look at the CC bonds, CC bond single bond, C double bond C, C triple bond C, the variation is so prominent. If we simply take these three, even then the molecules can show whether they have this particular moiety or functional group in their structure or not. So, part information can be obtained, and the beauty of this is that infrared radiation cannot cause electronic transition as the UV light or the visible light can do. So, what it does, it only plays around with the covalent bonds, stretching, bending and all kinds of vibrational modes, and by that itself that change will only occur if the infrared radiation of the matching frequency hits the molecule, not otherwise. So, this compatibility of the infrared radiation matching with the requirement of the bond, CC bond or any covalent bond, has to be similar. Only then vibrational modes will occur. The double and the triple bonds are stronger than a single bond and have correspondingly higher energies of vibration. Similarly, stretching modes have higher energies than bending modes of the same atoms. These vibrational modes can be excited to higher energy states, which cause the atoms to vibrate with great amplitude, that is greater displacement from the average position. So, you just imagine that there is a pendulum and it kind of goes as high as the energy, as high as the energization would take place. Vibrations can be excited by increasing the temperature or by absorption of photons of the appropriate energy. The energies of the vibrational modes are quantized and can be excited only with discrete amount of energy. A photon that has the same energy as vibration is said to be a resonance with that of vibration and can be absorbed. So, when it is in resonance with the requirement energy or vibration for a particular bond, or when it is matching or when it is compatible, you can say either of them, that is the time when it will affect the bond, and that affection or that implication will cause the absorption peaks to come in the spectrum. So, if I show you now an IR spectrum of natural indigo, you will clearly see there are certain portions marked with A, B, C and D, and these were found to be quite different from the synthetic indigo IR spectrum. Although in one go they both look alike, but if one looks at it very carefully, the regions of A, B, C, D marked in the synthetic indigo is quite different from the regions that are marked in the natural indigo A, B, C, D. So, that is how IR helps us to identify these molecules in a little more detailed methods, and you see all these peaks are nothing but absorption peak. It is the region between 4000 to 1500 which is of prime importance for an organic chemist to identify. The other region, which is from 1000 to 400, is the region which is the fingerprint region, which is of course for a very specialist to take care and understand these peaks. But the functional group area, that is from 4000 centimeter inverse to 1500 centimeter inverse, is where we can find the functional groups containing C bond, C double bond C, C triple bond C, C triple bond nitrogen, C double bond O, and so on and so forth. Now, let us come to another chromatographic technique which is very popularly used for dye molecules, high performance liquid chromatographic studies. The applicability of high performance liquid chromatography, that is HPLC, to analyze ancient textile dyes was first successfully demonstrated by Wooster in 1985. That is, it is not a very old technique, it is a very recent technique which has been applied to dye molecules. HPLC linear gradient elution method was first described by the analysis of indigoid dyes by Wooster and Verhoeven in 1991. Identification of blue and purple indigo dyes was also described using HPLC techniques by Wooster. Analysis of mangist, alizarin, turmeric, sandalwood, etc. were carried out much later in the year 1999 by Bhattacharya, and he also used the HPLC technique. So, you see that how useful it is for the identification of natural dyes and various different types of indigoid dyes, anthraquinoid dyes, and even turmeric and such dyes have been identified with the help of HPLC. Now, you see I have taken an example of a chromatogram. The graph that is generated from a chromatographic technique is called chromatogram, and the graph that is generated out of a spectroscopic method is called spectrum. So, that should be clear. Now, a chromatogram of synthetic indigo and a chromatogram of natural indigo has been shown here. You will see that there are small differences, but nevertheless one can make out that they are at least two different compounds, and the peak at 4.375 is very small in the case of synthetic indigo, whereas the peak at 4.85 is fairly large in the case of natural indigo, and that is because the iso indigo is also associated with indigo molecule. Therefore, these are very minute differences that one can figure out from the chromatographic technique. Now, when we are trying to look at chromatographic technique, there is one thing which is very important to tell you, and that is that one needs a standard, because it is a comparative analysis. whereas UV is an absolute analysis, IR is an absolute. You do not need another standard to compare. It gives functional group information, it gives chromophoric group information, and that is it. But in the case of chromatographic techniques, one needs to have a standard molecule of the same. Suppose if we want to evaluate the HPLC of alizarin in a Manjit sample. Now, in order to be able to identify alizarin in Manjis, we should have a pure sample of alizarin, and that is called as a standard. So, this kind of method is called a calibration method, and therefore, one needs to have very pure samples of these standards in order to identify dyes in mixtures. And in natural dye, there is also another problematic situation, because no dye comes as a single molecule. They are always 4 or 5 or maybe more dyes of similar structures. Structurally very little difference will be there, but nevertheless they are different entities. So, therefore, a standard is a must in the case of HPLC usage. Gas chromatography, one can use gas chromatography alone, or one can use it with the help of a mass spectrometric studies. Gas chromatography with mass spectrometer, that is GCMS, is an important detection method for natural products, providing chemical fingerprints for the peaks, from the peaks. Electron impact source EI and automated library searching makes chemical identification very easy. The gas chromatography serves a method to separate a mixture so that they enter the mass detector one at a time for identification. So, the chromatography actually only separates them, and the mass detector then starts detecting them and finding out whether the fragmentation pattern is of the known compound or it is an unknown compound or a different compound, and that can be done with the help of the library search. Now, here I would also like to emphasize that all organic molecules, whether they are dye molecules or they are colorless compounds, they all have a set fragmentation pattern, and therefore, the mass fragmentation pattern is a very characteristic of the particular compound itself. Two compounds on a gas chromatography can show the same retention time, but they may or may not be the same just based on the retention time. But when the mass fragmentation is checked and tallied, that is the ultimate determination whether the molecule is same or not. And therefore, it is one of the most serious kind of diagnostic test for identification of any dye molecule. Again and again I am telling that it can be applied for both synthetic and natural dyes. Now, if I have to show you a mass spectrum of quercetin, this is how it shows. The line shows here as the graph for different fragments that are broken from the parent molecule. So, the last line on the right hand side is the molecular ion peak, and then molecule breaks at the bonds which are more facile and gives daughter moieties, which then can be correlated to find the structure, and this is the mass fragmentation pattern of the dye called quercetin. It is a flavonoid dye. Similarly, I have taken another example of a dye which is called lycopene, and the molecular ion peak at 537, and there is a peak at 536 as well. So, the main molecular ion peak is 536 and M plus 1 is Sometimes such peaks arise due to the fragmentation pattern which appears at 537, and when 537 breaks, it gives a peak at 444, that is with the loss of 92 AMU atomic mass unit, and then subsequently a peak at 391 and then at 347 and at 321. These are the different fragment or daughter molecules that are deriving from the main lycopene. One thing you will see that the length of these peaks are different. That is arising because the stability factor of these fragments make the line longer. So, the relative abundance is higher for the ones which are more stable and which break after some time and not immediately. Nuclear magnetic resonance spectrometry. NMR is a preeminent technique for determining the structure of organic compounds. It is the only one method for which a complete analysis and interpretation of entire spectrum is normally expected. Of all the spectroscopic methods, this technique relies on the ability of atomic nuclei to behave like a small magnet and align themselves with an external magnetic field. When irradiated with a radio frequency signal, the nuclei in the molecule can change from being aligned with the magnetic field to identify and or elucidate detailed structural information about chemical compounds. NMR can differentiate between structural isomers and provide information about connectivity between atoms within the molecule. So, it gives proton NMR gives the number of hydrogen that are present, and the carbon NMR gives the number of carbon that are present, and the entire spectrum can be interpreted. Therefore, it gives a quite a discrete information about the structural details of a molecule, be it a organic molecule, be it a dye molecule. Coming to mass spectrometry, we did understand a bit of mass spectrometry when we were doing the GCMS part, but to be able to explain to you in a more detailed manner, because this is also one of the tools of identification of dye molecules. In order to measure the characteristics of individual molecules, a mass spectrometer converts them to ions, so that they can be moved about and manipulated by external electric and magnetic fields. Mass spectrometry is used to identify unknown compounds and quantify known compounds. It is sensitive and selective and is commonly used in combination with a separation technique such as gas or liquid chromatography to analyze complex mixtures. A mass spectrometer determines the mass of a molecule by measuring the mass to charge ratio, that is the m/z of its ion. Ions are generated by including either the loss or gain of a charge from a neutral species. Once formed, ions are electrostatically directed into the mass analyzer, where they are separated according to the m/z and finally detected. The result of molecular ionization, ion separation and ion detection is the spectrum that can provide molecular mass and even structural information. So, you see that this is an ultimate method for the determination of structure of an organic compound, and therefore, it is although called a spectrometric method, but it does not use any energy from the electromagnetic radiation region, unlike the UV, FTIR and the NMR, because in UV the UV light or visible light of the electromagnetic radiations were used. In FTIR, IR radiations were used. In NMR, the radio frequencies were being used, but this is one spectroscopic method where the molecule is an organic molecule which can be also a dye, is bombarded with an electronic beam, and then the molecule starts fragmenting. This fragmentation pattern is always the same, and therefore, it is like the only method of fragmentation of the molecule, and therefore, when the subsequent parts are formed, one can go back and calculate what has broken from where, and if a certain amount of AMU is broken, as what I had shown here in the case of lycopene, that 444 was generated from 536 or 537 only because there was a loss of a fragment of 92 AMU. So, this kind of correlation can only take place because these bonds break at a very particular time. So, we would now conclude that all these techniques are put together the tools for evaluation of or identification for dye stuff, which are nothing but organic molecules.